Hi everyone, welcome back. Numerous evidences suggest that the three-dimensional structure of a protein is the foundation of its biological function. Studies in modern structural biology often focus on molecular interactions. A molecule bound reversibly by a protein is called a ligand. A ligand may be any kind of molecule, including another protein. Binding of the ligands can lead to structural changes in the proteins. Specific conformational changes are frequently essential to a protein's function. In a multi-subunit protein, a conformational change in one subunit often affects the conformation of other subunits. In this section, we will take the myoglobin and the hemoglobin as examples to show how protein structure affects ligand bond, how structure changes in proteins affect their functions, and how proteins work. Myoglobin and hemoglobin may be the most studied and best understood proteins. They were the first proteins for which three-dimensional structures were determined. Both proteins are oxygen-binding proteins containing globins and heme prosthetic groups. Heme is present in myoglobin, hemoglobin, and many other proteins. Recall that heme consists of a protoporphyrin ring with a bound iron atom in its ferrous state. The iron atom has 6 coordination bonds, 4 to nitrogen atoms of the porphyrin ring and 2 perpendicular to the porphyrin. One of these is the binding site for oxygen. Iron in the ferrous state can bind oxygen reversibly. The globins are a widespread protein family, all having similar primary and tertiary structures. Most function in oxygen transport or storage. In humans and other mammals, the monomeric myoglobin stores oxygen and facilitates oxygen diffusion in muscle tissue, whereas the tetrameric hemoglobin is responsible for oxygen transport in the bloodstream. What's the reason for the difference between hemoglobin and myoglobin? Of course, it is because of their different structures. So next, let's talk about the structure characteristics and correlated functions of myoglobin and hemoglobin. Myoglobin is a single polypeptide of 153 amino acid residues with one molecule of heme. Thus, myoglobin has a single binding site for oxygen. As a typical globin polypeptide, myoglobin is made up of 8 alpha helical segments connected by bands, as shown in this figure. The helical segments are named A through H, which connected by bands, designated AB, CD, EF, FG, and so forth. The heme in myoglobin is bound in a pocket made up largely of the E and F helices, although amino acid residues from other segments also participate. An individual amino acid residue is designated either by its position in the amino acid sequence or by its location in the sequence of a particular alpha helical segment. For example, the histidia residue coordinated to the heme is his 93, which is also called his F8. Hemoglobin is a tetrameric protein containing 4 heme groups, one associated with each polypeptide chain. Thus, hemoglobin has 4 binding sites for oxygen. Adult hemoglobin contains 2 alpha chains and 2 beta chains. The three-dimensional structures of the 2 types of subunits are very similar. Furthermore, their structures are very similar to that of myoglobin, even though the amino acid sequences of the 3 polypeptides are identical at only 27 positions. Both myoglobin and hemoglobin can be reversibly bound to oxygen by heme. When a myoglobin or hemoglobin binds with oxygen, it's called oxygenated myoglobin or oxygenated hemoglobin. The ratio of oxygenated myoglobin hemoglobin to the total ones is called oxygen saturation, which is directly proportional to the concentration of oxygen. If we measure the oxygen saturation of hemoglobin and myoglobin at different partial pressure of oxygen, we can make the following graph. This graph is the oxygen binding curve of myoglobin and hemoglobin. The horizontal axis is partial pressure of oxygen. The vertical axis is oxygen saturation. We can see that the curve of myoglobin is a rectangular hyperbola, whereas the curve of hemoglobin is a sigmoid or S-shaped curve, quite different from that of myoglobin. Why are their curves so different? Maybe we can find the answer from the structure analysis of myoglobin and hemoglobin. Myoglobin is a single polypeptide. It has a very high affinity for oxygen and can bind quickly to oxygen even at very low partial pressure of oxygen. Therefore, myoglobin, with its hyperbolic binding curve for oxygen, is relatively insensitive to small changes in the concentration of dissolved oxygen, and so functions well as an oxygen storage protein in the tissues. However, hemoglobin, with its multiple subunits and oxygen binding sites, is better suited to oxygen transport. As we shall see, interactions among the subunits in hemoglobin cause conformational changes that alter the affinity of the proteins for oxygen. The modulation of oxygen binding allows the hemoglobin to respond to changes in oxygen demand by tissues. X-ray analysis has revealed that hemoglobin has two major conformations, the R state and the T state. T state is the predominant conformation of deoxyhemoglobin, which has a low affinity for oxygen and is not readily bonded to oxygen. The binding of oxygen to hemoglobin subunit in the T state triggers a change in conformation to the R state. In the R state, hemoglobin has a significantly higher affinity for oxygen. Therefore, hemoglobin undergoes a transition from a low affinity state to a high affinity state as more oxygen molecules are bound. As a result, hemoglobin has a S-shaped or sigmoid binding curves for oxygen. So what happens to hemoglobin during T to R transition? Oxygen binding to individual subunits of hemoglobin can alter the affinity for oxygen in adjacent subunits. The first molecule of oxygen binds weakly to a subunit in the T state. However, this binding leads to conformational changes that are communicated to adjacent subunits, making it easier for additional oxygen to bind. Then the T to R transition occurs more readily in a second subunit once oxygen is bound to the first subunit. The last oxygen binds to a subunit that is already in the R state, and hence it binds with much higher affinity than the first oxygen. This in fact that oxygen binding to individual subunits of hemoglobin can alter the affinity for oxygen in adjacent subunits is referred to as cooperativity. Therefore, binding of a ligand to one binding site of a multimeric protein can lead to conformational changes that affect the affinities of other binding sites. Max Pejos proposed that T to R transition is triggered by changes in the positions of key amino acid side chains surrounding the heme. In the T state, the porphyrin is slightly puckered, causing the heme ion to protrude somewhat on the his F8 side. As shown in this figure, the ion atom lies approximately 0.4 angstrom outside the porphyrin plane. Because iron is slightly too large to fit into the well-defined hole within the porphyrin ring. The binding of the oxygen molecule at the 6 coordination site of the iron atom substantially rearranges the electrons within the ion so that the ion becomes effectively smaller, allowing it to move into the plane of the porphyrin, which turns into a more planar conformation. The movement of the ion brings the proximal histidine and the attached F helix toward the porphyrin ring. These changes lead to adjustments in the ion pairs at the alpha-beta interface. In this process, some of the ion pairs that stabilize the T state are broken and some new ones are formed. Cooperative binding of a ligand to a multimeric protein can alter the conformation of this protein, such as we observe with the binding of oxygen to hemoglobin, thus resulting in functional changes. This effect is called allosteric effect. An allosteric protein is one in which the binding of a ligand to one site affects the binding properties of another site on the same protein. The ligands binding to the allosteric proteins that induce conformation changes are called allosteric effectors or modulators. The conformational changes induced by the modulators interconvert more active or less active forms of the protein. The modulators for allosteric proteins may be either inhibitors or activators. The binding of oxygen to one site of hemoglobin affects the affinities of other binding sites, therefore can be considered as both a ligand and an activator. There is only one binding site for oxygen on each subunit, so the allosteric effects giving rise to cooperativity are mediated by conformational changes transmitted from one subunit to another by subunit-subunit interactions. A sigmoid binding curve is diagnostic of cooperative binding. It permits a much more sensitive response to ligand concentration and is important to function of many multi-subunit proteins. So, interactions between a protein and its ligand is greatly affected by protein structure and is often accompanied by conformational changes, which in turn affects protein's function. Okay, in this section, we talk about the structural characteristics and the correlated functions of myoglobin and hemoglobin. The cooperative binding of the myoglobin subunits to oxygen makes it bind oxygen efficiently in the lungs, where the partial pressure of oxygen is high, and release oxygen in the tissues, where the partial pressure of oxygen is low. The high affinity of myoglobin for oxygen makes it fit for oxygen storage in the tissues. Our knowledge about the hemoglobin and myoglobin provide a deep understanding on correlation between conformation and the function of proteins. Well, that's all for this section. Thanks for joining. See you next time.